It is the most remarkable story ever told, a story written inside every one of us. Deep within each cell lies a code, a set of instructions exquisitely small yet profoundly powerful. This is your DNA. It's a biological language of just 4 letters, the blueprint that guided your formation from a single cell. It is the architect of your body and the keeper of your ancestral past. It dictates the color of your eyes, the texture of your hair, and the rhythm of your heart. Now, science allows us to read this code directly, unlocking secrets and beginning to answer the most fundamental questions about who we are. The blueprint of life is held within almost every cell. It's a library of books called chromosomes. We each have 46, organized into 23 pairs. The books are written in a 4-letter alphabet: A, T, C, and G. These letters form the rungs of the famous DNA double helix, where A always pairs with T and C with G. Specific stretches of these letters are our genes, like individual recipes. You inherit one set of 23 chromosomes from each parent, creating your own unique genetic blueprint. For every gene, you have 2 copies, 2 versions of the recipe. Most traits are influenced by thousands of genes working together, like a symphony played by thousands of musicians. This brings us to a fascinating question. If siblings share the same parents, why are they often so different from one another? The answer lies in a beautiful process of chance and shuffling. When their bodies create sperm or egg cells, only one copy from each pair is chosen to be passed on. Which copy gets chosen is completely random. But there is another layer of shuffling. The paired chromosome from grandma lines up next to the paired chromosome from grandpa and swaps pieces. This is called recombination. The result is a chromosome that is a mosaic, a brand new combination of your grandparents' DNA. This mechanism is the engine of diversity. It ensures that a population has a wide variety of traits, which is essential for adapting to a changing world. Sometimes, a change happens in the code itself. This is called a mutation. The word can sound alarming, but mutations are a natural and constant part of life. They are the ultimate source of all genetic variation. Mutation is not an error to be feared. It is the raw material of evolution. Our genes tell a story, but they do not write the ending. This is the risk equation. Our health is a product of our genes, our environment, our choices, and the element of chance. Your DNA might give you a certain predisposition. Think of it not as a sentence, but as a statistical nudge in one direction or another. Imagine 2 people with the exact same genetic variant that slightly increases their risk for heart disease. One person lives a sedentary lifestyle. The other person exercises regularly. Their genetic risk is identical, but their health outcomes will likely be vastly different. This is a profoundly hopeful message. Knowledge of a genetic risk can be empowering. It can motivate us to make healthier choices. It transforms genetics from a source of fear into a tool for prevention. This is known as a polygenic risk score. It can help identify those who might benefit most from early intervention or lifestyle changes, ushering in a new era of personalized preventive medicine. Now let us turn to the most complex and mysterious creation of the genetic code, the human brain. From the earliest moments in the womb, genes orchestrate a symphony of construction. They direct the formation of billions of nerve cells, guide neurons as they travel, and instruct neurons how to form trillions of connections. The brain's construction isn't finished at birth. Genes provide the initial scaffolding, but experience shapes the final structure. Neuroplasticity means your brain is constantly sculpted by life. Genes build the instrument. Experience is the musician, shaping the unique melody of your mind. Section 3. Chemistry of Mind and Mood. Our moods, our focus, and our ability to feel pleasure are all regulated by chemical messengers called neurotransmitters. The entire system of creating, releasing, and recycling these neurotransmitters is managed by instructions encoded in our genes. If a person inherits versions of genes that lead to, for instance, a less efficient serotonin system, their brain's chemical balance might be more easily tipped into a state of distress. The genes create a vulnerability, but it is often the interaction with life's challenges that determines the outcome. It helps us see these conditions not as a failure of character, but as legitimate medical conditions with a biological basis. It is a powerful shift from blame to biology, from judgment to compassion. We now understand that, at its very core, cancer is a disease of our genes. Cancer begins with one of our own cells, a cell that, through a series of unfortunate accidents, acquires mutations in its DNA. A normal cell follows a strict set of rules. Cancer cells are rebels. Their mutations have damaged the very genes that enforce these rules. A mutation might break the stop signal. Another might disable the repair machinery. A third might damage the self-destruct program. Cancer arises from a cascade of genetic breakdowns, turning a once cooperative cell into a rogue agent of chaos. For decades, our main weapons were chemotherapy and radiation. These treatments are powerful, but they are also blunt instruments. They attack all rapidly dividing cells in the body, both cancerous and healthy. The new way is to classify cancer by the specific mutations that are driving it. This insight has opened the door to a much smarter, more targeted way of fighting this ancient enemy. Instead of carpet bombing the body with chemotherapy, we can now develop drugs that act like smart missiles. It is a treatment tailored to the unique genetic signature of a person's cancer. These drugs can have remarkable effects. Within weeks, tumors that had spread throughout their body can begin to shrink and even disappear. However, cancer is a formidable and clever adversary. It evolves. The cancer learns to bypass the blockade. This is a constant battle, a biological arms race between our medical ingenuity and the cancer's ability to mutate and survive. Despite these challenges, the progress is undeniable. The story of cancer is being rewritten. For many patients, a diagnosis that was once a death sentence is becoming a chronic, manageable disease. Our genes aren't a fixed script. Life experiences add chemical marks that turn genes up or down without changing the DNA itself. This is epigenetics. But now, we can do more than just influence our genes. For the first time, we can edit the letters of life with a tool called CRISPR. It acts like a biological word processor, allowing scientists to correct harmful genetic mutations, offering hope for a true cure. But this power brings profound ethical questions. Where do we draw the line between treating disease and enhancing a human? Changes to the human germline would be hereditary, permanently altering our gene pool. These are not questions for scientists alone. We must proceed with courage and caution.